Assalamu alaikum, guys, and good evening. So today we are going to start last topic for this term: oxidative transport and electron transport and oxidative phosphorylation. This is the topic that is the part of introduction to metabolism, and you will be studying this topic again in detail in your next semester in 411. So we'll try to be as much simple as we can, but as part of the course content, we have to discuss this topic. So let's start the discussion. What is electron transport and what is oxidative phosphorylation? As you studied last time in the topic of digestion and absorption, we all need food, and the basic purpose of the food, number one, is to get energy. And secondly, we need the food to get the basic raw material like amino acids, sugars, to synthesize body's own required material. So, for the first case, when we require the food for energy, how that energy is obtained from the food, the answer to that is respiration, the process of respiration, in which we use oxygen to burn the food to get the energy out of it. So there are two levels of respiration. The one level of respiration is the one that we all know, that inhaling of the oxygen and then exhaling of the carbon dioxide. This is the normal concept of respiration. The second stage of the respiration, what we call the cellular respiration, is when we inhale the oxygen, it is going to be absorbed in the blood with the help of the hemoglobin, and then this oxygen is transported. This oxygen is transported to the cells, where it is going to burn the food, which we studied yesterday. After the digestion, the food is absorbed and then it's transported to the cells. So inside the cell, the food that is being absorbed is burned by the oxygen, and that is what we call the cellular respiration. And as a result of that cellular respiration, we get the energy out of it, and that energy is utilized by the body in order to perform different functions. The most important form of currency of the energy in the body is adenosine triphosphate, what we call the ATP. If someone is on unmute, please mute yourself. You should be careful about this. It's not the first class. So if someone is unmuted, it's disturbing the class. Please mute yourself. So oxidative phosphorylation actually is a process of the formation of the ATP, adenosine triphosphate. And this making of the ATP is linked with a process what we call the electron transport. So that's why collectively we call this topic as electron transport and oxidative phosphorylation. Okay, so let's go in the detail. In the cell, there are two levels of compartmentalizations. There is one what we call the cytoplasm, and there is the other one that's called the nucleus. Now, within the cytoplasm, we have certain membrane-bound organelles, and one of the most important membrane-bound organelle within the cytoplasm is mitochondria. And you all have studied about mitochondria. This is called the powerhouse of the cell. So the energy that I was talking about, that we produce from the burning of the food, is actually produced within the mitochondria. So mitochondria is actually the site where the energy production takes place. In simple, we can say that mitochondria is the site where the ATP production takes place, because as I mentioned earlier, that ATP, adenosine triphosphate, is the ready made currency of the energy for us. So ATP are produced within the mitochondria, or in other words, the process of oxidative phosphorylation takes place within mitochondria. So, in order to go through in detail about this process, it is important for us to know a little bit about mitochondria. Now, mitochondria is a membrane bound organelle, and it has got two layers, one outer membrane, which is relatively permeable to most of the stuff, and then comes an inner membrane, which permits only very specific transport molecules. So that means there are two layers or two membranes of the mitochondria, the outer membrane, which is relatively permeable, and the inner membrane, which is selectively permeable. It only allows certain things to pass through it for which they need transporters. Now, this inner membrane is actually impermeable to many things, which include these two molecules, NADH and FADH2. And the inner membrane is permeable to pyruvate. So I've just mentioned that inner membrane is permeable to pyruvate, which is one of the intermediate compound within the cell metabolism, and it is not permeable to two molecules, NADH and FADH2. What is NADH and what is FADH2? NADH and FADH2 are referred to as universal electron carriers. These are the molecules which carry electrons. And you will study about them in a bit of more detail in the metabolism when you study BCH 411 in your next semester. Here in this presentation, I'm going to explain a little bit about NADH and FADH2, and then you can consult from the book as well. This NADH is called nicotinamide dihydroxide, and FADH is flavin adenine dihydroxide. These are the two molecules which I said are the universal carriers of electron. They carry electron. They can take up electron and they can lose electron. So that's why they are actually carrier or transporter of electrons. Now, in case of the mitochondria, we have to understand that whatever metabolism takes place within the cell, this compartmentalization play a very crucial role. There are certain metabolic pathways which take place within the cytoplasm, and there are certain metabolic pathways which takes place within the mitochondria. Keeping in mind that mitochondrial membrane is not permeable to everything. So that means if something is going on within mitochondria, that means it is separated from the reactions which are going on in cytoplasm. So in this case, the glycolysis, which is the 10-step reaction of conversion of glucose to pyruvate, takes place in cytoplasm, while the Krebs cycle, which is also be called the TCA cycle, and beta oxidation, which we are going to discuss right now, they takes place in the mitochondrial matrix. What is mitochondrial matrix? Mitochondrial matrix is the matrix which is present between the outer and the inner membrane of the mitochondria. So it is to be understood that the glycolysis, which is the first part of the metabolism, takes place in cytoplasm. The glycolysis is then followed by the Krebs cycle, which is also called as the TCA cycle. That takes place in the mitochondrial matrix, which is between the inner and outer mitochondrial membrane. And then the beta oxidation, what we call the oxidation process, that also takes place in mitochondrial matrix. Now, the energy which is coming out of the food as a result of the burning mainly comes from the redox reaction. What are redox reaction? I'm sure that you guys have studied in your general chemistry. Redox reaction is a term used for the coupled reaction of reduction and oxidation. Because whenever there is a reduction reaction going on, simultaneously there must be oxidation reaction going on as well. Because these two reactions are coupled reactions. Similarly, if you remember, you have studied about endergonic and exergonic reaction. which in general chemistry you call exothermic and endothermic. So if some reactions result in the release of energy, the other reactions will result in the absorption of the energy. In a very similar way, redox reactions are if one reaction results with a reduction, the other side of the reaction goes with oxidation. So this is what we call the coupled reaction. Now in glycolysis, which is going place in cytosol or cytoplasm, there is production of two NADH molecules. Then from pyruvate to the next product, which is being catalyzed by pyruvate dehydrogenase reaction, that takes place in mitochondrial matrix, there are two molecules of NADH produced per molecule of the glucose. And in the Krebs cycle, what we call the TCA cycle, which is also going on in the mitochondrial matrix, you have the molecule of 6 NADH and 2 FADH2 per glucose. So now let's summarize that upon the complete oxidation of one molecule of the glucose, you got 2 NADH as a result of the glycolysis in cytoplasm, 2 NADH as a result of the pyruvate dehydrogenase reaction going on in mitochondrial matrix, and 6 NADH and 2 FADH2 per molecule in the Krebs cycle that takes place in mitochondrial matrix. So let me summarize that with the one molecule of the glucose, there are 2 NADH produced in cytoplasm and 8 molecules of NADH are produced in the mitochondrial matrix, and 2 molecules of FADH2 are produced in the mitochondrial matrix. So now there are 10 NADH and 2 FADH2 molecule produced per molecule of the glucose. Remember that all of them are produced outside the mitochondria. So in the first slide, I told you that inner mitochondrial membrane is impermeable to both NADH and FADH2. So in order to bring those NADH and FADH2 within the mitochondria, you need some sort of process, and that is what we are going to discuss in detail, and that is what we call the electron transport chain. So let's summarize it again, that for the synthesis of the ATP, which takes place within the mitochondria, you have to transport these NADH and FADH2 inside the mitochondria in order to get the energy out of it. And in order to transport them in the mitochondria, you need a mechanism, and that mechanism is being called as electron transport chain. So that's what we start now, electron transport chain. Now this electron transport chain is a group of proteins which are embedded in the inner mitochondrial membrane. These group of proteins are called as redox proteins. So let me revive again. Redox proteins are the proteins which will be simultaneously reduced and oxidized. That's why we call them redox proteins. And as I said, that their location is on the inner mitochondrial membrane. The purpose of these redox proteins is actually these are the binding sites for the NADH and FADH2, and thereby they facilitate the binding of NADH and FADH2 to transfer their electrons from mitochondrial matrix to the inner side of the mitochondria, where the oxidative phosphorylation takes place. So these redox proteins, they have binding sites by which the NADH and FADH2 can attach. This attachment is on the side of the matrix, and as a result of this attachment, the NADH and FADH2, which I refer to electron carriers because they carry electrons, they transfer their electrons. And as a result of that, the NADH is converted to NAD positive and FADH2 is converted to FAD. Once they lose their electron, they become NAD positive and FAD. And these electrons will be taken up by other molecules, which will be, then transferring those electrons to the oxidative phosphorylation process, which results in the synthesis of ATP molecule. So these redox proteins, which I mentioned here, they are 4 complexes. Now these four complexes, they are by nature protein, and they transfer two electrons in a specific order. The proteins are localized in the complexes and they are embedded in the membrane, as I said. The electrons ultimately end up reducing oxygen to water. So the end product of this process is water. We all know the equation of respiration, and the end product of the respiration is carbon dioxide and water. So this end product of the water, it actually comes from the oxygen, because oxygen after gaining electron will be reduced to water, and that's what we call the process of respiration is completed. Now this is electron transport chain picture. It might not be big enough to understand, but you can go through this later on to enlarge this and see. But just to let you know that this is called complex 1, this is complex 2, this is complex 3, and this is complex 4. So these are the four complexes. This is mitochondrial matrix. This is outer side of that, and this is inner side. That means this is outer membrane of the mitochondria, this is inner membrane of the mitochondria, and this is the mitochondrial matrix. So one can say that this is the outer side of the mitochondria and this is the inner side of the mitochondria. So some of the NADH and FADH2 are in the cytoplasm, if you remember, and some of the NADH and FADH2 are within the matrix. So these complexes, as I said earlier, they have got binding sites for NADH and FADH2. So you can see the complex 1 can bind with NADH, and NADH after losing its electron will be converted to NAD positive. And these electrons which are taken up by complex 1 will then result in the pumping of the four protons inside the membrane of mitochondria. So that means the energy of the electron is being transformed in pumping electrons from there to the inner side. Similarly, this complex is, itself gaining electron, and NADH is losing electron. That's what we call the redox reaction. We have another complex here which is specifically for FADH2, and you see that FADH2 can lose its electron and become oxidized to FAD, while this complex will pump two electrons out of this. So as a result of the electrons of the NADH, two electrons results in the pumping of four protons, while as a result of the FADH, the two electrons will result in the pumping of two more protons, but it will be carried on in the next complex. You can see that this transfer to this one. So this is the complex 2, and that pumps out four protons. So we have four protons pumped as a result of two electrons each here. Then we have the complex 3, where you can see that these two electrons will be taken up by this complex 3 or 4 to finally pump two more protons, and the oxygen is being converted into water, which is the final step of the respiration. And in the last step, we have a protein called ATP synthetase. Now, these are the four complexes, and this is separate protein. This is called ATP synthetase. The name itself indicates that it is required for ATP synthesis. And these protons which are being pumped out during this electron transfer, now they will come inside. And as a result of this protons pumping inside, the energy is released, and that energy is used to produce ATP from adenosine diphosphate to ATP adenosine triphosphate. Now, I know that it's a little bit tricky and little bit in detail, but as I said that we are not going to discuss this topic in much of the detail, because that's the part of the discussion that you will do again in the BCH 411. But let me summarize for your understanding that what actually happens. First thing is that we have universal electron carriers in the cell which accept electron, and they are able to transport electron. These universal carriers are NADH and FADH2. Now, Mitochondria has got two membranes, the outer membrane and the inner membrane. The outer membrane is relatively permeable, while the inner membrane is very, very selective. It doesn't allow NADH and FADH to pass through it. Now, in order to produce ATP, because the ATP production takes place within the mitochondria, we need those electrons to be transported inside. And for this, there is a mechanism called electron transport chain. This electron transport chain is the combination of complex proteins which are embedded on the membrane. They have the binding sites for NADH and FADH2. The binding of the NADH and FADH2 on those complexes will transfer the electron, as you have seen there. Binding of NADH transfers the electron to complex 1, and the NADH is converted to NAD positive. This binding of electron results in pumping of 4 protons from this side to this side, right? This is what we call against the concentration gradient. Then these 2 electrons are transferred to another complex CEQ, and this complex is linked with another protein where FADH2 can also lose its electron, so 2 more electrons coming here. Then these 2 electrons are being then transferred to complex number 3, and as a result of this electron transfer, 4 more protons are being pumped out. Then these electrons are transferred via cytochrome C to complex number 4, and as a result of this electron transfer, 2 more protons are being pumped out, and finally these electrons are being transferred to oxygen, and as a result of that, oxygen is converted to water. So actually what's going on, NADH is losing its electron to complex 1, complex 1 is giving its electron to complex 2, complex 2 is giving its electron to complex 3, and finally complex 3 is giving its electron to complex 4. That's why we called it a redox reaction, because one is losing electron and the other one is gaining electron. So that means one is oxidized and the other one is reduced. And as a result of this continuous oxidation reduction process, what's going on? Protons are being pumped out, and they are being pumped out against the concentration gradient. That means from higher concentration to lower concentration is a normal process, but against the concentration gradient means from lower concentration to higher concentration. So once it's transferred its electron to group 2, it becomes available again for binding of another NADH, and so on and so forth. Finally, the electrons are being taken up by oxygen, and then oxygen is then reduced to water. So oxygen is gaining electron at the terminal stage, and it's finally reduced to water. And because of this whole process, the protons are being pumped out, so it creates proton gradient in this place, and now these protons will come inside again through the ATP synthetase. And as a result of this energy is released, and that energy is utilized for making adenosine triphosphate ATP from ADP and inorganic phosphate. That's how the oxidative phosphorylation takes place, and that's how the ATPs are produced. So with this, it's pretty much complete, but I'll quickly go through the presentation. I'm not going in further detail, but just to let you know the further details. Here, this is the group complex 1, which has already been shown in the picture. The details are there. It has NADH binding site, and it pumped 4 protons. So 2 points are important for you at this stage, that complex 1 has a binding site for NADH and it will pump 4 protons. Then complex 2 has a binding site for FAD and it pumps no protons at this stage. Then complex 2 will transfer electron to complex 3, and as a result of this, 8 protons are pumped out per NADH and 4 protons are pumped out with 1 FADH. Remember, FADH has a low energy than NADH. And then finally complex 4, where the complex 4 is going to transfer the electron to oxygen, you can see here, and then oxygen is then reduced to water. With this, we can say the electron transport chain is complete. And as a result of this, total 10 protons are being pumped out per molecule of NADH and 6 protons are being pumped out per molecule of FADH2. And finally, as I said, that these protons will come inside, and as a result of that, because the energy is released, and that energy is utilized by the mitochondria to produce ATP from ADP and inorganic phosphate. So that's how the process takes place. Approximately 4 protons are equivalent to 1 molecule of ATP. So in this case, you can see here, if they are, let's say, 16 protons, so roughly saying that there are 4 ATPs produced as a result of this. So this is the formula calculation. Uh, NADH one molecule carries roughly 10 protons, so approximately equivalent to 2.5 ATP, and one FADH2 will bring about 6 protons, which are equivalent to 1.5 ATP. So this is the exact calculation that one molecule of NADH is equivalent to the energy of 2.5 ATP, and one FADH is equivalent to the production of 1.5 ATP. So this detail is not important for you at this stage. You will study that again, as I said, in 411. But just to let you know, in pyruvate dehydrogenase there was one NADH, so 2.5 ATPs. In Krebs cycle, there are 3 NADH produced. You will go through in detail in your next semester. So there will be 7.5 ATPs. One FADH is produced in Krebs cycle, so that is 1.5 ATP. And one GTP is produced, which is equivalent to ATP. So if you altogether total per molecule of the glucose, there will be 25 ATPs produced. So this is what I'm not going in further detail. I'll stop here. These slides are not related to you, so I'm not going to discuss about them. But I'll be finishing it up at this point, where you have just idea that how ATPs are produced and a little bit about the calculation that how much total ATPs are produced from one molecule of the glucose. So you must go through this calculation that one molecule of glucose gives you about 25 ATPs. Why? Because this calculation, which is being shown here, this calculation is with one molecule of pyruvate, and a glucose molecule gives you two pyruvates. So one pyruvate gives you 12.5. So two pyruvates will give you 25. So that's why we said that one molecule of glucose upon complete oxidation gives you 25 molecules of ATP. So we'll stop here. And with this, we have completed our course for this term. And inshallah, you will have enough time to go through the content. And if you have got any question, you can come back anytime and ask the question. There is no problem. And inshallah, we'll be seeing you on Wednesday for finalizing our lab manual. And in this class, I'm going to discuss about the uh, some details about exam pattern and other related stuff. So thank you very much. I'm going to share the recording once it is available. Thank you for that.